Good afternoon, everyone. My name is Irene García. I'm a PhD student at the Translational Immunology group, and today I'm going to present my work titled Heterogeneous IL-9 Production by Skin-tropic and non-tropic memory T cells in atopic dermatitis. Atopic dermatitis, from now on AD, it's an heterogeneous chronic inflammatory skin disease. Clinically unaffected skin is characterized by epidermal barrier defect and reduced microbiota diversity. Most of moderate to severe AD patients are colonized by Staphylococcus aureus species and present an impaired immune response, characterized by a core Th2 signature that induces an intense pruritus, together with IgE sensitization to aeroallergens and the participation of other immune responses such as Th1, Th17, and Th22. In this study, we will focus on IL-9 response. IL-9, it's a cytokine produced by a wide range of immune cells, but its major source seems to be CD4 T cells. The cytokine binds IL-9 receptor for signal transduction and in the skin is expressed in immune cells, keratinocytes, and melanocytes. IL-9 participates in allergy as it plays a pleiotropic role in Th2-mediated inflammatory diseases such as asthma and food allergy. Likewise, it plays a pathogenic role in atopic dermatitis as is increased in serum as well as in lesional and non-lesional skin. Altogether correlates with disease severity and serum levels of total IgE and CCL17, which is the most reliable biomarker for atopic dermatitis. Moreover, single nucleotide polymorphisms in IL-9 and IL-9 receptors have been associated with high increased risk of suffering from atopic dermatitis. CLA stands for cutaneous lymphocyte-associated antigen, which is an adhesive molecule present in more than 90% of T cells infiltrating the skin, but it's nearly absent in T cells infiltrating other non-cutaneous tissues. These T cells are primarily expressed by CD4 and CD8 memory T cells, although they are also present in other immune cells. Immunohistochemical studies have shown that these T cells abundantly infiltrate AD skin compared to normal skin. And this is possible because these T cells have the capacity of homing the skin, as CLA, together with other adhesive molecules, participate in the transmigration process. Once these cells are within the skin, they can become activated by antigen-presenting cells presenting, for example, allergens such as Hasdas mite or Staphylococcus aureus toxins such as staphylococcal enterotoxin B, which are AD triggers. Then these T cells have the capacity of dehoming the skin and recirculating back to the peripheral blood, where they can be easily isolated, making them a powerful tool to translationally study skin diseases such as atopic dermatitis. Currently, IL-9 production is thought to be transient and selectively produced by skin-tropic memory T cells. Additionally, no differences in IL-9 producing CD4 and CD8 skin-tropic and non-tropic memory T cells have been observed between AD patients and control subjects. It's worth mentioning that all these studies have been performed under a specific polyclonal activation. Moreover, it remains unexplored whether pathological triggers of AD, such as Hasdas mite or SEP, can induce IL-9 production. For this reason, the aim of this study was to explore IL-9 immune response in moderate to severe patients through the Hasdas mite and SEP activation of both skin-tropic and non-tropic memory T cells in the presence of autologous epidermal cells. To do so, we recruited 52 non-treated moderate to severe adult patients and 17 control subjects. From blood, we isolate plasma that we use to measure Hasdas mite and Staphylococcus aureus specific as well as total IgE levels. From peripheral blood mononuclear cells, after several rounds of immunomagnetic separation, we obtain CLA positive and negative memory cells. Lesional skin biopsies were enzymatic and mechanically disassociated until we obtained an epidermal cell suspension that was co-cultured together with skin-tropic and non-tropic memory T cells in the presence or the absence of Hasdas-Mite and CEP activation. After a period of activation, in the case of CEP 24 hours or the Hasdas-Mite for 5 days, we collect the cell supernatant and we measure several AD-related mediators. Then this immune response was correlated with the clinical data collected from each patient. Now let's start with the results. Hasdas-Mite induced IL-9 production in both CLA positive and CLA negative memory T cells co-cultured with epidermal cells in AD patients, but not in control subjects. Next, we proposed to characterize our model, and we saw that IL-9 was produced in a time-dependent manner, that T cells on their own are able to produce IL-9, but this production is increased in the presence of autologous epidermal cell. A Transwell assay revealed that not only the presence of epidermal cells was necessary, but they needed also to be physically in contact with T cells. Then, the neutralization of class 1 and class 2 molecules drastically reduced IL-9 production, which means that it depends on HLA-mediated presentation. The polyclonal activation of our co-cultures induced CLA positive and CLA negative IL-9 production, but we did not see the preferential CLA positive IL-9 production as observed under Hasdas-Mite stimulation. IL-9 was simultaneously measured with other AD-related mediators in our co-cultures, and we find that it was the most abundant cytokine produced in both co-cultures upon Hasdas-Mite stimulation. Moreover, this production was correlated with clinical data of our patients, and we found a direct correlation with both Hasdas-Mite specific and total IgE levels. Next, IL-9 production was not uniform among all of our AD cohort, which allow us to stratify patients into IL-9 producers and non-producers. We then compared between these two groups the clinical data and found that these patients, the IL-9 producers, have increased specific IgE and total IgE levels, which mean that these patients exhibit a higher degree of allergen sensitization as well as an allergic status. Moreover, they present a higher pro-inflammatory state, as all the cytokines measured in our co-cultures were also increased in these IL-9 producing patients compared to the non-producing patients. Next, we wanted to know if other known triggers of atopic dermatitis could induce IL-9 production. CEP was able to induce this IL-9 production in both, again, CLA positive and CLA negative memory T cells co-cultured with autologous epidermal cells. But this production was lower compared to other Th1, Th2, and Th17 cytokines measured in our co-culture, which means that CEP, it mildly induces IL-9 production as compared to Hasdas-Mite allergen. This production also was heterogeneous and allowed us to stratify patients again into IL-9 producers and non-producers. And these patients, the IL-9 producers, also had a higher pro-inflammatory state, and they were more exposed to Staphylococcus aureus, as we find more IgE-specific levels to Staphylococcus aureus in these patients. So, to sum up, IL-9 is produced in a heterogeneous, non-temporally manner by skin-tropic and extracutaneous T cells in response to both Hasdas-Mite and CEP. Hasdas-Mite is a potent inductor of IL-9 production. Also, IL-9 correlates with both total and specific IgE levels. AD patients with IL-9 production exhibit increased degree of allergen sensitization, allergic status, and a higher pro-inflammatory state. For this reason, the conclusion of this study is that allergen sensitization reflects the heterogeneous IL-9 production in vitro, which may allow patient stratification from a clinically homogeneous population. Thank you. I would like to thank the Catalan Society for Immunology for giving me the opportunity to present my work, and also to the audience for your attention.
So, any question from the audience? Anybody? No? Will you listen to me, or it's okay? Yeah? So, it was very clear. In fact, I don't have any question to do, but what is the next point? Are you going to try to use this for the clinical use? Are you going to use as a biomarker? What are you group doing right now?
Well, we are trying to understand the disease, because it's a highly heterogeneous disease. We have seen that atopic dermatitis, for example, one of the best treatments that it's dupilumab, actually, that it's a target therapy against IL-4 receptor alpha. It only works in 30% of the patients. So we are trying to describe and better know the different responses that take place in this disease in order to better characterize or to endotype these patients for for a clinical use to a more target therapy at the end. Yes.
The aim is to use this as a biomarker for a target-specific marker. Okay.
Mm-hmm. It's possible. Any question from the audience? No? First of all, Irene, well done. Molt bonic el treball. And a question. This work is performed with multidisciplinary with dermatologists.
Yes.
And I am allergist, but we are friends. But my question is, it has been surprising for this dermatology group to know that this stimulus with house dust mite has been superior than Staphylococcus aureus, because you know that there is some controversial about the role of IgE allergy in atopic dermatitis, and I think that these works explain, at least in part, the role of the IgE sensitization in patients with atopic dermatitis. What has been the discussion in the group, please?
In fact, it's funny because it's true that we work with dermatologists, and they are actually surprised because they think that Staphylococcus aureus is more important than these allergens. But we are also going to start working with allergologists from a hospital in Malaga, and they are going to give us the other version. And also we would like to implement new triggers, for example, or cat or other allergens, for example, because they are also important and highly present in atopic dermatitis patients. So, yeah, it's...
Okay, thank you very much.
Thank you.